Assalamualaikum and good day everyone. I'm glad to meet you guys today. So in this session, we are going to cover chapter 9, reproduction and developments, focusing on subtopic 9.3 and 9.5. For subtopic 9.3, there are 4 learning outcomes that you should be able to understand. The first one, you have to define what is the embryogenesis, and after that, state developmental stages from zygote to the formation of morula, blastocyst and gastrula through cleavage, and then define what is the organogenesis and state the organ form from each germ layers during organogenesis. Before we discuss detail about the content for subtopic 9.3, let's recap the definition of fertilization. Fertilization is referred to the unions of two gametes, right? It's referred to union of haploid male gamete and female gamete to form zygote. Alright, so now we proceed to the definition of embryogenesis. What is the embryogenesis? Embryogenesis refers to a process by which embryo is formed and developed. Fertilization occurs in the fallopian tube and also known as oviduct. And after fertilization, the fertilized egg or zygote starts traveling down to the fallopian tube towards the uterus. During the movement from fallopian tube towards the uterus, zygote undergoes cleavage and differentiation. Cleavage is referred to the rapid mitotic division. Zygote undergo first division and become two cells about 24 hours after fertilization and complete about 30 hours after fertilization. I hope you still remember about the concept of mitosis, which is a process to increase the number of cells. Then the two cells continue second mitotic division, become 4 cells. Then after third division, 8 cells form with 2 tiers of 4 cells. Third division complete in about 70 to 70 hours after fertilization. Through cleavage, a fertilized zygote become a morula and blastocyst. The morula travel to uterus around 3 to 4 days after fertilization. Then when a fluid-filled space called blastocyst appear, morula will become a blastocyst. This diagram shows the differences between morula and blastocyst. The second process involved during fetal development is organogenesis. Organogenesis refers to the process which formation of organ from 3 germs layer. After implantation complete, cell then undergo process called gastrulation and produce 3 germs layer. The 3 germs layer are ectoderm, mesoderm and endoderm. These 3 germs layers of embryo developed into various organs of the organism. For this part, you have to remember the example of organ developed from each germs layers. If you realize the fetal development process actually is the beginning of our journey as a human being. Now, let's take a look at subtopic 9.5, growth. In this subtopic, we are focusing on definition of growth. Growth is the irreversible permanent increase in quantitative parameters that can be measured. In this subtopic, you will learn about human growth curve and growth pattern. Growth pattern include limited growth, unlimited growth, and allometric growth. More details about this subtopic we'll discuss in tutorial class. Thank you for your attention.